this all visible?
Yeah, brilliant. Thank you.
Perfect. Okay. Hello, everyone. Good evening on this Monday evening, and thank you for the invitation for this Nature History Live talks. And thanks, Holly and Kieran, for organizing this. So yeah, my name is Katharina, and I'm a wildlife veterinarian and a researcher at the Zoological Society of London at the Institute of Zoology, working for the Garden Wildlife Health Project. And today I want to tell you a little bit about citizen science and how we can use it to monitor the health of wildlife, and in this case, the wildlife we find in our gardens. Okay, it didn't swap. There we go. Okay, so why do we even bother? Why study wildlife health and wildlife diseases? What's the purpose and what do we actually do with this information? So in general, we have three main objectives, which is to monitor what's already here. So endemic diseases, those that are known to be present in the country and are affecting wildlife. Also to detect new diseases or diseases that we haven't seen for a while or weren't aware of the presence in the country. And to be prepared, meaning to perform horizon scanning. So keeping an eye on the horizon for diseases that haven't yet arrived in the UK, but have the potential to have an impact if they were to arrive. And we do all of this with the view to conserve biodiversity and protect wild animal welfare, but also to safeguard public and companion animal health as well as livestock health, and also to document any potential wildlife crime and help prevent any persecution. So what is citizen science? We mentioned that a lot. So citizen science, when you put that into the good old Google machine, you will come up with lots of different definitions of citizen science. But what does it all mean? In general, in summary, it means that it is scientific work, for example, any data collection that is undertaken by the members of the general public, often in collaboration with or under direct supervision of professional scientists or scientific institutions. So when we talk about wildlife disease surveillance schemes, so they have in general the goal to detect a range of infectious or non-infectious disease threats and are typically reporter-led, and therefore they rely on soliciting reports of observations of sick or dead wildlife for their investigation. And citizen science offers a very powerful tool to achieve this form of surveillance, and it's particularly well suited to any charismatic wildlife species that is frequently observed and positively perceived by the public. So there are many advantages to citizen science, to the public, but also to science, of course. So for example, if you participate in citizen science, you will yourself gain a certain level of scientific education, and participation encourages engagement with nature and therefore promotes your general well-being. And you also participate in public health improvements as well as in optimizing habitat management for the well-being of wildlife as well as ourselves. But of course, there's also huge benefits to science because this allows us to collect large-scale data over long periods of time and across huge spatial distances. And especially when we think about private gardens, as we as scientists would never be able to monitor what's going on in the gardens without the help of the public, as we obviously can't be there ourselves. So the Garden Wildlife Health Project is an example for such a citizen science scheme. And our fundamental aim is to monitor the health of, but also identify any potential disease threats to garden wildlife. But there are, of course, many more aspects to it. For example, once we do identify a disease threat to British wildlife, be it in a single species or a whole taxonomic group, we are also trying to be actively involved in trying to find and then promote ways of mitigating that threat wherever appropriate, of course. And this can vary in scale from providing individual members of the public with advice on what they can do in their gardens to reduce disease spread, up to developing national mitigation strategies to deal, for example, with any invasive species or new pathogens. So the species that we focus on within this project are garden birds and birds of prey, amphibians, reptiles, and also the hedgehog. So whilst the project is led by ZSL, where we also do our disease investigations, and we work in very close partnership with the British Trust for Ornithology, the BTO, FrogLife, and the Royal Society for the Protection of Birds, to draw from their knowledge on the natural history of the species we perform this surveillance on, but also to benefit from the wide supporter and membership bases and their authoritative voices in terms of all their public outreach. And in particular, with regards to the BTO, their proven track record with citizen science is basically the foundation of our project, as you will hear in a bit. Of course, in order to learn as much as possible and to reach as many people as possible, we collaborate wildly with many other organizations from a range of sectors, including governmental organizations, conservation NGOs, wildlife gardening organizations, rehab centers, academic institutions, and so on. So how does the project now actually work? So here's a very brief schematic introduction into what we do, which I will explain then in more detail in the following slides. But with the help of the public, we undertake scanning surveillance for infectious as well as non-infectious diseases that are affecting wildlife in British gardens. To facilitate the study of the diseases we might identify, we build a sample archive and collaborate with a wide range of organizations and integrate our findings with population monitoring in order to determine any potential impact on a population level. And this allows us then ideally to provide people with evidence-based advice on disease mitigation, habitat management, and other best practice strategies. So let's have a bit of a closer look on how this works. So there are, of course, many different methods for conducting wildlife disease surveillance. But we in general, as mentioned, rely on the public to report diseases like disease incidents or fatalities of garden wildlife to us via our website. And occasionally, when a freshly dead carcass is involved, we can also obtain the specimen for postmortem examination. So we receive these disease reports in two different ways, through opportunistic reports, and secondly, through the so-called systematic reports. So what does that actually mean? So briefly, the opportunistic reports. Opportunistic reporting happens ad hoc, meaning that members of the public report any sightings of sickness or death to us via our website, which is shown here. And when you click that button, report sickness or death, takes you a few minutes to create a profile, and you can then log reports as and when necessary ad hoc. There is a quick form that you fill in where you can add details of the species that you see, the number of animals that are affected, any potential signs you've observed of illness. And there's also a section to add any additional queries or comments, and also to upload photos, which often proves really helpful. And if a freshly dead carcass is available, there will be a box that you can tick, which will prompt one of the project veterinarians, so me or my colleagues, to contact the reporter regarding the possibility of a submission to us for postmortem examinations, which is under strict veterinary instructions, and all costs are always covered by the project, so it's free to participate. So as I mentioned, these opportunistic reports happen as and when you observe something, meaning we receive them when they occur, which means that we gain a maximum potential observer coverage, and this also offers the best way to potentially detect any novel incidents or conditions. But of course, as so many things, this also has some disadvantages or issues. For example, there is a potential bias associated with observer effort, meaning that you will, for example, likely receive more reports from areas with a denser population or more gardens. There's also bias towards certain conditions that might have just received a lot of press coverage, as people will suddenly be much more aware of a certain condition and what it looks like, or they will even try and actively spot it in their gardens, like, for example, avian poxvirus infection here in this article, which can cause these typical tumor-like growths, especially in tit species, and is therefore very easily spotted. There's also often species bias, meaning that more charismatic species are more likely to get reported than less popular species. Other types of bias could include incidences of unknown causes, as people are, of course, less likely to report a case where the cause of death seems obvious to them. For example, cat predation or acute trauma like a window collision or a road traffic accident. However, we need to be aware that there remains the possibility of an underlying infectious or non-infectious condition that might have predisposed the animal to such a traumatic event in the first place. Therefore, even reports of such obvious causes can be very, very valuable. Opportunistic reporting occasionally also means that people report after the event has actually happened, which means that we can't obtain any potential carcasses should they be involved, and we might lose some valuable data. And importantly, with opportunistic reporting always concerning individual or isolated cases, there remains an uncertainty around the impact that any suspected or ideally detected condition might have on the species on a population level. So overall, we can conclude that whilst opportunistic reporting is extremely valuable and provides us with lots of hugely important information, there remains a certain lack of statistic validity to the data that we can actually collect, and therefore we're limited with some of the conclusions that we can draw from them. So because of these limitations, we need a different system. So we also use a network of so-called systematic reporters, consisting of over 15,000 garden birdwatch participants, which are citizen scientists spread across the whole country, which is organized and led by the British Trust of Ornithology. So the garden birdwatch participants, they report on a weekly basis, which is incredible, regarding the presence or absence of certain different species in their garden. And this contributes towards abundance data. And whilst they're reporting these species, they can include any information on sightings of sickness or death, or they can indicate that there are no signs of ill health observed, as this is also extremely valuable to us and very different from I haven't looked. So all this provides us with some statistical validity, and our disease surveillance combined with this population data now provides insights into any potential impact a disease or a pathogen might have on the species on a population level. I also wanted to briefly mention that as a response to the national lockdown that we're all experiencing and people spending more time in their gardens, the BTO Garden Birdwatch, who are also celebrating their 25th anniversary this year, they made the participation of this scheme free of charge for a one-year membership. So I thought I'll share this in case this is of interest to any one of you or somebody you know. So this making the scheme free led to over 10,000 users in the past 12 months and over 6,500 regular reporters, meaning that they post more than 10 reports and have therefore doubled the numbers that we received from last year through the scheme, which is really great. Okay, so whether opportunistically or systematically, the members of the public report to us via our website or the BTO website, which allows us to collate all necessary data that we require for disease surveillance. And wherever possible, we try to assign a so-called syndromic incident definition. That means a suspected diagnosis, which is based on set definitions that we have developed over the years of surveillance, but which of course we can't confirm without performing an actual examination on the animal. So these suspect diagnoses often relate to diseases that are easily identifiable by the clinical signs and species or the species that might be involved. For example, here to the left, we have avian poxvirus infection again, which causes these typical tumor-like growths on the head or body of the bird. And are easily spotted, or on the right, where you can see a chaffinch affected with the species typical chaffinch lesions, which are caused by either papillomavirus or a parasitic mite infection, or possibly even a combination of both. As I mentioned before, when reporting to us, there's also the option to submit photos with the report, which often proves quite useful, as for example, something that might be observed as a growth on the head could turn out to be a tick, as seen in this case, or the other way around. Assigning these suspected diagnosis is of course not always straightforward. So the more information we receive, the more likely we can categorize and understand what might be going on in the specific garden or in the country overall. For example, here a fluffed up bird, this blue tit, that doesn't tell us anything other than a bird feeling unwell or even cold. So it's very unspecific. However, a bird being fluffed up with food stuck to the beak, showing signs of difficulty swallowing whilst trying to feed and being of a relevant species like the greenfinch in this picture, that would be an indication for the bird being affected by Finch trichomonosis, which is a parasitic disease, which I will explain in more detail later on. So whilst these suspect disease definitions give us a lot of important information, in order to be certain of what the cause of disease or death was, we need to perform a postmortem examination. So if the report involves a dead animal, and should the carcass be considered to be in a suitable condition for such an examination, there is the option to offer the animal for submission. So in this case, we contact the reporter directly and explain the submission process in detail, including any potential public health precautions that might have to be considered and how we will proceed, and providing a special delivery label. And again, the costs for all of this are carried by the project. So during these postmortem examinations, we conduct a full gross examination of the carcass and all the organs and body systems in our labs at the Institute at ZSL. We also perform additional diagnostics, which almost always include microbiology and parasitology. And then, depending on the species or the findings, we conduct further tests like molecular diagnostics or histopathology or radiography in some cases. But the overall aim of all of these examinations and the postmortem examination overall is to determine a cause of death, as well as also identifying any potential underlying or other conditions that might have been affecting the animal but might not have directly contributed to its death. And additionally, we also want to gather general data on morphometrics and find out what is actually normal in a certain species. For example, what pathogens can be found in the given species without causing disease, as lots of these animals are still quite understudied species. So these maps give you an idea of the coverage that we have achieved using these two different systems, this opportunistic and systematic reports. And as you can see, we have received reports from of sick and dead wildlife from more or less the whole of Great Britain. And the gaps that you see reflect likely the bias whereby you're less likely to receive reports from areas where humans and wildlife habitats don't overlap that much. So we use these reports and postmortem examination findings to identify and study the infectious and non-infectious diseases affecting British wildlife. And the postmortem examination enables us to conduct extensive sampling of tissues from the specimens that we examine. And over the last 25 years, we have therefore created one of the largest wildlife tissue archives in Europe. And this archive acts as an invaluable resource to facilitate our studies. Like, for example, if we were to detect a disease or a new pathogen, this archive enables us to go back and through the previous cases and to see if this specific pathogen or disease might have been around before already, but we have potentially overlooked it. And if it was, then for how long and what happened. So this helps us to identify emergence events of diseases and to also trace the spreads of the disease or pathogen in time and space. So when working with citizen science, we think it's absolutely essential that this is a two-way street. So this has to be a give and take, basically. So when we receive reports or specimens for examinations, we always communicate back to the reporters. We provide them with information on what we think might be causing the observed signs, or also what was the cause of death if we found out, any related potential public health information or concerns that could be relevant, and if possible, any advice on disease mitigation, habitat management, wherever applicable. So one way of providing such information and evidence-based advice is through our disease fact sheets, which are available on our fact sheet library on our website. And they contain lots of information on the pathogen causing a certain disease, the species involved, the signs this might cause, as well as any public health concerns, and again, advice on potential mitigation if applicable. Here's an example of such a fact sheet on lungworms in hedgehogs, and they're also all free to download as PDFs. Of course, we also try to publish any new findings in scientific journals, and wherever possible, we aim to publish those in open access journals to make our research available to the wider public. And they're all accessible or at least listed on our website. We also provide an interactive map on our website, which allows everyone to look into the spatial distribution and also in the number of certain diseases or species that we observe. And here is a brief example where I mapped all the disease incident reports, which are the blue circles, and the postmortem examinations, which are the blue triangles, that we received involving hedgehogs from 2 consecutive years, 2018 compared to 19. However, you can map and compare up to, I think, 4 maps and look into species and distributions and have a look what happens around you in your area or all over the country. In addition to advice communicated to the general public, we also provide information and advice to inform NGO and government decision making. So one example for this is our participation in the Great Britain Wildlife Disease Surveillance Partnership, which convenes twice a year to discuss any current or potential future disease threats to British wildlife, and also whether any potential action should be taken. And between all the organizations within this partnership, we cover the vast majority of all our native species, which is great in avoiding any overlap or any duplication of effort. We're also active on Twitter and Facebook. Social media is key these days, where we aim to share any recent findings or seasonal advice, but we also share news from partner organizations or collaborators or any other related information. We also provide our reporters with biannual e-newsletters, so electronic newsletters that get emailed out, which offer up-to-date summaries of our research findings, public outreach events that we participated in or are planning to attend, or any other relevant project updates or useful information. When possible, in order to be more visible to different groups and audiences, we try to get involved in various outreach events in order to communicate and interact with a variety of people. And we also try to collaborate on articles or press releases with any nature, wildlife magazines, or other organizations where possible. Okay, so I would now like to give you 2 brief examples of how Garden Wildlife Health Project worked, like how it helped us inform about specific conditions, basically. So one example will focus on a specific disease, which was detected in garden birds and how we learned about its effects, and the other to show how we're trying to gain an understanding of if there is a certain conservation issue present. In this case, I'm going to use the hedgehog population decline as an example. So first, the Finch trichomonosis. So Finch trichomonosis, I mentioned it briefly before, is a parasitic disease caused by a protozoal, meaning a microscopically unicellular small parasite called Trichomonas gallinae. The disease predominantly affects greenfinches and to a lesser extent also chaffinches, but it has been reported in most finch species and also many other garden birds to date. This infectious disease causes severe lesions in the gullet of the bird, leading to obstruction of this gullet, and therefore basically preventing the affected bird from being able to swallow. The bird, which still will attempt to feed, will now have to drop the seed again, which is covered in saliva and now contains the parasite, and therefore offering an easy way for the next bird to get infected when it comes to pick that seed up. So affected birds, which again mostly greenfinches and chaffinches, will typically appear fluffed up and lethargic, which are the general signs of ill health, but they then tend to be observed trying and failing to feed, often with food and saliva stuck to the beak and the plumage around the faces. So yeah, as I mentioned, this disease can easily get transmitted via contaminated food when the birds feed together, but also through like bird-to-bird feeding during the breeding season or when adults feed the youngs. So this disease was first identified in British finches in 2006, and it raised a lot of public concern given the large number of sudden death in greenfinches and to a lesser extent chaffinches, as you can see here in this photo, where there's on the very right a chaffinch lying there. And this disease is also one of the best examples of an emerging infectious disease, meaning a disease that wasn't previously present in a certain area or species. And it has had a dramatic impact on greenfinches in Great Britain. And so far, as you can see, its incursion in 2006, it has been suggested that since 2006, this Finch trichomonosis has led to a population decline of about 70% in the breeding population in greenfinches, which is an incredible impact on a population level. However, although the disease has only relatively recently emerged in British finches, it is important to note that the pathogen, or the parasite in this case itself, is not actually new. So it has been well documented and known for hundreds of years as a cause of ill health and also death in columbiforms, which is pigeons and doves, and also in birds of prey that occasionally feed on them. And whilst we can't, of course, be absolutely sure about this, it seems to fit with the theory that in the human adapted landscape, and in this case, particularly the popular practice of garden bird feeding, which of course has lots of positive effects for the bird populations, species are brought together that would normally have very little interaction, and which likely provided the parasite with the opportunity to jump species and affect a new host, in this case with a devastating impact. And now once the disease is established within a new host, the parasite can continue to circulate within the species when they live and feed together. So in case of garden birds being affected by this disease, whilst we can't treat animals in the wild, there are measures that we can take to help reduce further disease transmission in garden habitats. So we therefore always recommend, where multiple birds seem to be affected, to temporarily stop feeding during the disease outbreak, and to always follow good feeding station hygiene. Of course, to always use fresh foods from reputable resources, and if possible, it's always a good idea to rotate feeder positions to avoid accumulation of waste food or any bird droppings, and of course, fresh water, ideally on a daily basis, is always a good idea. All these things, of course, rely on us being watchful and trying to raise awareness regarding hygiene precautions, not only for the well-being of wildlife, but also for ourselves. Okay, now I very briefly give you some insights into our hedgehog health investigations. So this is the UK's only spiny mammal, and the only mammal that is included in our project. Hedgehogs are regularly voted the country's favorite wild mammal, and unfortunately, despite its popularity, no matter which way we look at it, each of these lines represent a different survey using different techniques for estimating the national hedgehog populations. British hedgehogs have been rapidly declining over the last 25 years. And it's now estimated that we have lost at least a third, so over 30%, maybe even up to half of our hedgehog population. And hedgehogs have recently been listed as a vulnerable species by the IUCN. So our current understanding is that this is likely to be a multifactorial issue, including things like habitat loss, therefore habitat fragmentation, and leading to road traffic accidents. And we, within the Garden Wildlife Health Project, we're trying to look into whether any disease might be a contributing factor to this problem that the hedgehogs are facing. So there is already a lot of knowledge on hedgehog health from individual animals, which tend to get commonly presented as wildlife casualties, rehab centers, or wildlife hospitals. But there remains an information gap regarding the diseases that affect hedgehogs in the wild, and whether they could be contributing to this observed population decline. So whilst we do find a variety of infectious diseases affecting hedgehogs, and in varying degrees, we very commonly find non-infectious conditions to be the ultimate cause of death of these animals. Most commonly things like mammal predation or road traffic accidents. There's also still only very limited information on how certain toxins might be affecting our hedgehogs. So we are therefore collecting samples to further investigate, for example, whether rodenticides or slug pellets might be cause for disease or even death in hedgehogs. And we have now created a large sample storage for toxicological investigation. So what do we know about hedgehogs so far? Unfortunately, not that much, but we have learned that non-infectious causes of death are very common, but also that hedgehogs do carry a variety of different infectious conditions. Some of these might even have a zoonotic potential, meaning that they can also affect humans or even also other pets, which is why it's also always important to follow good personal hygiene precautions like wearing gloves when handling hedgehogs, or also any equipment like food bowls that were in direct or indirect contact with the animal, and to always thoroughly wash your hands afterwards. We need to further investigate how our use of pesticides, rodenticides, and slug pellets might affect their overall health or even their population levels. And to date, and that's really important, there is no evidence of a single disease condition affecting our hedgehogs on a population level, so we couldn't identify that. So to summarize, what have we learned about citizen science and wildlife health surveillance? So citizen science offers a powerful tool to conduct large-scale disease surveillance. And because diseases are highly dynamic and often unpredictable, they require close monitoring over longer periods of time in order for us to differentiate between an endemic, meaning a well-established, and an emerging, meaning a new disease. And whilst we gain a lot of information from opportunistic, meaning ad hoc reports, in order to understand whether a condition might have a population level impact, we need to combine those with systematic data. Sample archives are crucial to facilitate the study of pathogens or diseases in order to be able to trace them in space and time. And finally, especially when working with the public, collaboration and communication are the key to success when working with citizen science. So yeah, I'd quickly like to thank, of course, my colleagues, Becky and Joe, and our partner organizations, the WTO, FrogLife, ZSL, and the RSPB, and of course, our funders. And I thought I leave you with some information on future events that could be of interest to you, one coming up in January, which will focus on pathological investigations, including a session on how we conduct examinations within the Garden Wildlife Health Project. Looking closer into reptiles and snake fungal disease. And then there will be another great talk on citizen science and how it supports wildlife and conservation, and that will take place in February. Okay, I hope I could give you some insights into how our project works and how we can learn a lot when we work together and collaborate wildly. And now, fingers crossed, I will be able to answer any potential question you might have.
That was great, thank you so much. We've got a few questions in already, so I think I'll dive in now. I'm just going to stop sharing your screen, then if everyone wants to turn their cameras back on, it'd be nice to see you all again. Right, yeah, I'll jump into it. So Lewis sent in a question earlier asking, if we see a finch that's obviously suffering, can we do anything to perhaps catch it to prevent the spread?
So if it's a single isolated case and it's easy and you can approach the bird in a way that you don't put the bird itself or yourself at risk, then we say that if it's that unwell, then you can take it carefully, wearing gloves ideally, washing your hands afterwards thoroughly, of course. You can take it and carefully place it, for example, in a little carton box and take it to a wildlife center or a vet to have a look at it. But yeah, this only works if it's really a single isolated case, because if it's a disease that's already occurring in your garden, then it's more about stopping the spread and the transmission of disease. Yeah, so that's a case-to-case decision.
Great, thank you. Graham has sent in a question asking, is it worth reporting any animal deaths, even if the disease may not be obvious?
Yes, absolutely. As I said before, that we learned, so we can only deal with these species, but there are other citizen science projects that deal with other species. But we most of the time don't know the cause of death, which is why we want to hear and learn as much as possible about where it is, what was observed, and especially when we don't know what it is, those are the more interesting cases, because we want to understand, because it must have died of something, what has actually happened. And that's when postmortem examinations come in very, very valuable. If the carcass is available and fresh, and we can organize submission, which of course in the current times is especially tricky. But under normal circumstances, a fresh and intact carcass, we could organize submission and then try and find out what actually happened.
That's great, thank you. We've got a question from Jan as well. She was saying, this year I've done my dormice surveys wearing face masks and gloves and avoiding handling the dormice. How likely is it that COVID-19 could infect wild animals, and particularly dormice?
That is a very good question, and I'm definitely not the right person to be asked. There is, I mean, we know that there's a lot of diseases in wildlife that can jump to us, and the other way around, zoonotic diseases. We know that there is many suggestions that COVID originates from wildlife, but there are some studies that look into what animals can potentially carry it, but they're investigated mostly by DEFRA and other organizations, so we are not involved in that. But I'm sure that you can find lots of information about this, for example, on the DEFRA website, so I would recommend having a look there.
Well, thank you. Dan is asking, has your work on the Garden Wildlife Health Project shed any light on the decline in house sparrow populations?
So we have collaborated recently with other organizations on a study on avian malaria, which is caused by Plasmodium, which is a disease that's in blood cells. And so there is some indication that, for example, avian malaria has contributed to the current decline in house sparrows. Whether that's the only cause or something else is going on is not something we know as of yet, but we know that this is a condition that has contributed to the population struggle that we are observing in house sparrows. And you can, if you go to our website, gardenwildlifehealth.org, we have a great disease fact sheet that has in all little bit more detail everything described about avian malaria if you're interested in learning a bit more about this. But we're still looking into this, because yeah, it's a disease that we know is present, but we are trying to understand the impacts on certain bird populations.
Great, that sort of leads on to the next question, which is from Joe. He was asking, are you working on bird/avian flu, and how quickly can you track and trace diseases?
Also a great question. So we are not working on avian flu. So avian influenza has been around for, like, it's here every year, and there's different strains, which is like it changes from year to year, and it's back now, and I know it's all over the news, but it's mostly affecting like poultry, and in wild birds, the animals that we see are affected mostly are water birds like geese and swans. And again, there is really good information about this on the DEFRA website that can tell you all about what's happening in the wild bird populations and how quickly we can. I'm always surprised how quickly we manage to do things if we suddenly find the right thing to do. Some things take longer, some things go quicker, and this is why archives and things like that are so essential.
Great, thank you. Gordon is asking, what would be the process if I found a dead hedgehog?
Okay, so the process if you find a dead hedgehog would be that ideally you go to a website and you report it, and if it is in a, so if it's not in a fresh condition, you just report what you've seen, where you've seen it. So we need the date, the signs you've seen, and the location. And if it's freshly dead and it's intact, meaning there are no open wounds and there's no smell to it and no fly activity, then you can tick a little box that says that you would like to submit it for examination for us to find out the cause of death, which will prompt an email with us. So we will call you and we will discuss the case, because we need to make absolutely sure that we all understand how this works. And then with special packaging instructions and a special label, we can then submit it actually via the Royal Mail following all these guidelines. And then we will conduct the postmortem examination as and when it arrives. And then we will update the reporter with all our findings with the full postmortem report, in an ideal case with a cause of death, but sometimes we don't know why it died, but we will inform you of what we found.
That's brilliant, thank you. We've got a question from Fiona about hedgehogs as well.
Mm-hmm.
She's asking, is there any evidence of high parasite loads in young hedgehogs that are found underweight towards the end of summer?
That is a very specific question. So hedgehogs carry so many pathogens and parasites that often don't seem to do anything to them. However, there are, of course, for example, typically in weak younger animals in summer, we often see intense lungworm infections, which are two different species of worms that can affect the lungs of the hedgehogs, but they're very often also carried without causing disease. So it's always a question, is something else sort of suppressing the immune system, and then the lungworms have the opportunity to flare up and actually cause severe problems to the breathing system? Or is it that it's a high infestation of the parasite that caused the disease in the first place? But it's very difficult to say, but we definitely do find a museum of parasites in those creatures sometimes, yeah. But they can cope with a lot. It's incredible.
Great, thank you. Yeah, Fiona added a comment in the chat saying it's a theory that her local hedgehog rescue centre has put forward.
Okay, well, I'd be interested in that theory. She can email us if she wants to. You find the email address on our website, would be interesting.
Right, thank you. Stephen's asking, if looking at conserving overall biodiversity, is there ever a motive to collect data in order to conserve certain native parasites themselves?
Also a good question. I wouldn't know, but I mean, we know that this all biodiversity is all, I mean, it's a big system. We need everything and everyone to work together, basically. So it's a good question, actually. If a certain, like for example, the hedgehog declines and the parasites it carries, whether they would then also have a population issue. I've never thought about it, I have to admit. Good question, thank you.
Yeah, the other way around, isn't it?
Yeah.
Mark was asking, is there any evidence of immunity in finch populations?
I guess that's regarding the Finch trichomonosis. Yeah. That is also a very difficult one. So we see sort of waves. So there are years where it's worse and years where it's better. But does that mean that the finches get used to the parasites, or does it mean that there are less finches around? I mean, when you lose 70%, then the spread, of course, will slow down because you don't have that many birds around anymore. So I think that's where the long-term monitoring is the essential thing that comes in. So hopefully we will learn about this a bit more in the next years. But it's still one of the most common things that we find in these species and one of the most commonly diagnosed disease within our project. But we definitely see lesser cases than we might have 5 to 10 years ago. But why that exactly is, I can't say yet.
Thank you. Peter is asking, how do you track wildlife health in species that are migratory?
You work together with other institutions, collaborate wildly across Europe, for example. But I mean, those are especially interesting when it comes to disease surveillance, because they do go all over the place, right? So, for example, with Finch trichomonosis, after it has started in 2006 in the UK, we have then started to see it in Scandinavia, southern Scandinavia, and then it started to appear also all over mainland Europe. And the question was, how is this happening? Because green finches are not known to be migratory species, right? So, and then looking into this, we learned that the chaffinches were also affected. They go in the summers to southern Scandinavia to breed there. And on the way back, they're much lazier. They don't go all the way back up. They go slowly across the coasts of Europe, mainland Europe. So it's an interesting one, yeah, but you need definitely to collaborate.
Definitely, thank you. Maggie is asking, if I send a report of a dead hedgehog to Suffolk Wildlife Trust, does the Garden Wildlife Health Project pick this information up? Does it come through to you?
No, it doesn't. So they would have to tell us specifically. But if you report, like you can report one species to different schemes. So the only reports that really go through to us are from the Garden Birdwatch. So when there is, so not the species abundance data is what we collaborate on with them, but the reports that actually go to our system is the health information that are submitted through the Garden Birdwatch. And of course, our other, like Frog Life and the RSPB, whenever there is a disease incident reported to them, they alert us about it. They encourage the reports be done to our website directly. So if you can and are willing, then we're happy if you were to report directly to our website.
Great, thank you. And we'll send around the link to the website afterwards as well for anyone that hasn't had a chance to scribble it down quickly. We've got a question from Stephen here. You mentioned conservation and animal welfare as aims at the beginning of the talk. Do you ever find that these areas conflict with one another? For example, diseases that might be infecting common birds considered pest species that are outcompeting / over predating on rare species?
Intense questions. I don't know. That's a tricky question, I think. So I mean, I think it's a lot of it is in a balance of things, as in we can, I think so we can all only do what we find is correct in our own gardens. So we interfere with transmission the way. So it's also the question, should I stop feeding if I have 2 sick birds in my garden in winter, or would it, like, what outweighs, where's the benefit? Should I stop feeding not to risk transmission, but do the birds need the energy from the food? So I think it's all a lot of personal decision making in many aspects, and a way of balance. We cannot avoid disease. Disease has always been present. What we try to avoid is it having a huge impact on certain species. And of course, if that costs another species, then we need to rethink our approach.
Great, thank you. Okay, I think that's all the questions from the chat. If anyone else has got any, I would suggest sending them out. Well, 2 have just appeared right now. Maggie has said, oh no, so it's just a comment from Maggie, sorry. Sharing of information with other conservation bodies, I think, is an opportunity missed. This sharing of info could help save wildlife. And then Arlene asked, the infection that affects the feet of chaffinches, could you repeat the name, please, for her?
So the chaffinch-like lesions, they're mostly seen in chaffinches and to a lesser extent in bullfinches, also greenfinches. We have recently also found them in 2 or 3 cases in dunnocks. So they're caused either by Fringella papillomavirus, so it's a papillomavirus, or by a mite infection called Cnemidocoptus, that's a mouthful. So it's a mite infestation. And very often we find that it's a combination of both. And this is normally not affecting the overall health of the bird in any way or form. But of course, it can lead to these growths being very heavy, so impairing their ability to fly, perch, sometimes predispose them to entanglement or predation. But yeah, also we have on our website a fact sheet on it where you can find all these beautiful names, pathogens involved.
Yeah, you're doing well to remember them all and pronounce them correctly. I think I'd get tongue-tied with all of those.
It's part of my job, I guess.
Yeah. Yeah, I think, oh, there we go, there's one more. How important is citizen science as a contributor to conservation, sort of as a whole?
I think massively important, and I think it's getting more and more important. So science has its limits, and if we don't work together, we can't overcome these limits, especially when we, like in the example of Garden Wildlife Health, which I mentioned briefly at the beginning, is in what way could we know what's happening around feeding stations in private gardens? The amount of people who would have, no one would be happy if we knock on your door and be like, can I sit in your garden for 5 hours and observe your birds? So we need people to look at it. And also when you go for walk, so we're not limited just to gardens. So if you're in a park or somewhere and you find something, observe something, as long as you know the location roughly, that's also really helpful, because it's not possible without volunteers to gain this information about the whole population, what's happening. And I think it's going to be more and more popular and relevant in many aspects. Yeah, I think when it comes to conservation, collaboration and working together is and will be more and more key.
Definitely.
Yeah.
Right. Well,
great ending.
It was, yeah, good question to end on.
Yeah.
So, yeah, thank you very much for joining us this evening. That was